Hi.
Hi.
Hello, thank you for joining me.
You're welcome. Thank you.
We'll be starting in just a couple minutes to make sure that people can get in.
Okay.
Okay, dokie. Well, we will go ahead and get started. I think a lot of people are like doing Valentine's Day stuff, so I don't blame them. I'm gonna have a pretty like a couple people asked me to go ahead and record myself talking about a couple of specific things for review. Ling Ling, I would just wanted to ask you, since you're the only one live with me, is there anything specifically you wanted to talk about before I jump into the heat of it?
Probably no, I don't know. So I think I'm independent. So usually I just review by myself.
Okay. All right. Well, I'm gonna just go ahead and jump into a few things that people asked me to do. So first, we wanted to talk about selective versus differential. Oh, hi, Daniela. So we wanted to talk first about differential versus selective media. And so I'm going to go back to like my little pool party example, right? So the selective portion of selective media is going to select the things it wants to grow, right? So MSA plates, they're going to grow halophiles. They're selecting for the halophiles. So when you are getting ready for a pool party, you're going to select the people that you want to come with you, right? Or select the people that you want to attend. So that's going to be our selective portion of our media. And then when we turn to the differential portion, it's like when we finally get to the pool party, we're going to play a water gun fight, right? we're going to differentiate the two teams. So one's going to be team yellow and one's going to be team red, right? So if we look at the MSA plate, the pink or red one is going to be our non-mannitol fermenting, and then our yellow is going to be the mannitol fermenters, right? So that's kind of the rundown on those. We will need to know what makes up each of the plates and what makes them selective versus differential. The only two that you're going to be responsible for in terms of like everything you need to know about is the MSA versus the EMB. You'll also need to know what is our one that's strictly differential. What is the one plate that is strictly differential? Does anybody know? You can take a shot at it in the chat. You can say it out loud. But does anybody have an idea? Yes, you would be right, Hannah. Our blood agar plate, right? So our blood agar plate just is differential for and yes, I will flowchart the 5 kingdoms, but that is going to be our strictly differential plate. So that's just like no matter where you are, there's 2 groups of people, right? It's going to grow everything, but it's going to differentiate them. Let's see. So I just wanted to go over really quickly the 5 kingdoms. So I had a pretty good picture, Daniela, that I sent you that Dr. B approved. So the one thing with the kingdoms is that everything's going to fall into one of these kingdoms. So you have Animalia, you have, and that's just us, right? We're in Animalia. There's going to be things like fish. There's going to be things like, I just threw my pen. There's going to be things like rats. There's going to be dogs, right? We're going to have a ton of animals in that section, mammals or not. Marsupials, if you will be in that one. So that is going to be our Animalia kingdom. Next, we have Plantae, and Plantae is going to be anything that's a plant. It's pretty straightforward for those two. Fungi, they're fun guys, you know. They are multicellular, and unlike plants, their cell walls aren't made of cellulose. What is our special little thing that's in fungi? I'm gonna bring up the PowerPoint. There you go, chitin. Chitins are fun little one. So that's, I think, I'm gonna pull up the PowerPoint just to make sure that I'm giving you guys all the right stuff. I swear I've downloaded like all of these like 15 times. So let me not download it for another time. And then share my screen. I'm just gonna share my screen in general. That way you guys can just see where I'm going. Let's see. So you guys also, Microsoft 365, get out of here. If I can close it, please. There's like, you guys can't see it, but there's like my bar, and then I can't click in the bar or else I'll click something. So that little X was like right underneath it. Anyways, so I guess I should have started a little bit more broad. Okay. So let me draw it out behind me. I'm gonna get in a comfortable, I'm trying to mark where the edge of the board is so I'm not like writing off. Okay, you guys can see it in those parameters. So everything's going to be either prokaryote or eukaryote. Okay. So one of the main differences between these two is that prokaryotes, they're going to do binary fission. Eukaryotes, we're a eukaryote, right? So I always like to think of it like my toe, like I have toes, right? Not for free, but I have toes. And where was I going with this? I got very sidetracked. My train of thought was gone. But plants don't have toes, right? We have toes, but they don't have toes. And or like bacteria doesn't have toes. So I just don't want to confuse you guys. So eukaryotes, mitosis, prokaryotes, binary fission. Okay, so part of prokaryotic characteristics, just think of bacteria versus us, right? If we have it, and you can say, because you guys have studied anatomy and physiology at least a little bit, you guys know what eukaryotes are. We have DNA, we have a nuclear membrane, right? We're very complex cells. We have lots of organelles, but we don't have peptidoglycan. But prokaryotes are very, they're a lot more plain. So they don't have a nuclear membrane. They don't have a nucleus. They don't have many organelles. The only organelles that they really do have, I guess you could say, they're ribosomes, if they're technically considered an organelle, but they're not membrane enclosed. They have peptidoglycan. Obviously, we saw the difference between gram-positive and gram-negative. I guess I could add that right here. So, gram-negative, gram-positive. We got that. But yeah. So let's move on just a little bit. Do you guys kind of understand this portion, the pathogen portion? About what makes each one, what makes each one, like, why is it hard to fight a eukaryotic pathogen?
Structures in the eukaryotic pathogens are similar to our own cells, so you can't really target things like the peptidoglycan cell wall formation.
Yeah,
like
exactly right. Yep. So with bacterial pathogens, their makeup is so different from us that we can use that to our advantage and target them. That way we can, lyse the cell wall, because what do we not have that they have? A cell wall. We can lyse that and pick on things in that cell wall, and it won't harm us. It might, make us feel a little, not so great, but it's going to that selective toxicity is what we want. Let's see. Yeah, you guys understand that there's layers. So this is kind of what we're going to outline. Okay. So just to continue just a little bit, because I know that sometimes it's confusing. Here, let me include this in our little flow chart, right? So we're going to look at eukaryotic cell walls first. So part of the 5 kingdoms that is eukaryotic includes Plantae. And I know it's really confusing too, because we were taught plant versus animal cells a lot. So now we're going eukaryotic versus prokaryotic. So let me try to clear the water here. So obviously we're going to have a eukaryotic animalia. We're going to have Plantae. We're going to have fungi. Yes, fungi. Okay. So those are our 3 that are going to be in our eukaryotic. And because all of these are multicellular. Yes, all of these are multicellular. So then all we have left here is plus prokaryotes. I know that they have special sciencey names, but that's kind of our division so far. So for their cell walls, let me see how I'm going to do this. For their cell walls, so I'm going to just do CW, and I'm going to do it straight across. So for our, sorry, I have a chat. Let me double check. Which one would be Archaea or Protozoa? So Archaea are a special type of, let me flip to that slide. So Archaea, I want to say prokaryote. I just don't want to tell you guys the wrong thing. I have another chart. So in terms of the 5 kingdoms, I think you guys just really need to know that there are 5 and what exactly are their characteristics. It's not really going to go into too much detail. It's basically if you copy down the slides or if you decide to print them out, then you can fill in the blanks with whatever she talks about with. But I don't think that it's going to go too deep into the whole, you know, and it's so specific in terms of the 5 kingdoms. So let me try to get back on track. Oh, I'm sorry. It's a little haywire right now because I was asked outside of class to include a couple things. And I want to make sure that everybody's questions are answered because I know some of you guys won't be able to go to a session before your exam. So I'm just trying to get that for you. So Archaea would technically be part of this because their structures are part of eukaryotes because their structures are the most similar to eukaryotes. So Archaea would be here. Protozoa will be over here. Protists are part of Protozoa. Okay, I hope I answered that question relatively well.
Yes, thank you.
No problem. Okay, so let me just kind of breeze through these real quick. So for the sake of time, I'm sorry, I'm not going to go into too much detail. Just like if I had to take anything away from these slides, take away this, like I would say eukaryotic cell walls and then write this down. And if you understand everything, great. Understand the glycocalyx, but for both eukaryotic and prokaryotic. So one of the biggest things is that people mix up eukaryotic versus prokaryotic. I'd say like 6 times out of 10, so like the majority of the time, there is going to be a question about prokaryotic organisms rather than eukaryotic, right? We're talking about microorganisms, which are more often than not going to be prokaryotic. A few exceptions would be things like parasites that are eukaryotic, right? So I would just kind of try to write this out and fill it in. So like things about the cell wall, glycocalyx, flagella. I'll ask you guys this question. Does bacteria have cilia? Good, I see no's. Good, good, good. There you guys go. You guys know about taxes. And just so you guys know, Tuesday class, you guys are gonna go to your review session. and you guys are going to, or you're going to go to class, and it's going to be just one basic review session. And if you couldn't make today, like if you were gone from class, I would recommend trying to go on Tuesday. If you tell Dr. B, hey, I was absent on Monday, can I just sit in on this review session for Tuesday, Thursday? It's the same exact time, it's just on Tuesday. And I believe that there will be an open lab followed like today. So she will outline basically everything that's going to be on lab and most of the stuff that's in lecture. She said to me especially that people are going to be probably more stressed out about lab. And because of that, they're going to neglect the lecture portion. So if you guys are stressing out about lab, but you have been able to do all the labs pretty well, you got the technique down, you feel okay, focus on lecture. Lecture is where it's going to hurt the most, and you'll need that information in order to make an in-depth decision about some of the questions, because they do require a little bit of critical thinking. So if you kind of have the idea about what flagella is and what cilia is, you might not do so well in the lecture portion, but if you know how to gram stain like down to a T, you can do it in your sleep, then you will have no problem on the lab. So that's the best advice I have right now, is probably just going back through the lecture. Let's see. Yeah, it's basically, if you can take this down and categorize it or put it into like two separate things. So if you wanted to do a eukaryotic packet and a prokaryotic packet, do that. And like take your notes and just group them like that. It will help. Let me see. If you guys can just look through these and be like, I understand it, that's good. I don't know if you guys have any specific questions about this, because let me look back in the chat. I feel like I'm missing something or I'm not answering something specific. I think I'm also going to send a picture on Remind so that you guys can kind of see what the 5 kingdoms are. Like basically there's one picture that sums it up, and that's basically all you need to know in terms of the 5 kingdom material. So I would just do that. I promise I'm more organized than this. Okay. So I sent you guys a little picture. That'll help with the 5 kingdoms. And you'll just need to know other physical structures. Eukaryotes, a little less in depth, but prokaryotes is where it's at. This is what you need to know, mostly, I will say. But I'm not going to say that there's no questions about eukaryotes, because there very well could be. So remember, what are the 2 glycocalyx that bacteria can have? Let's see. Okay. There you go. So slime layer and capsule are what are our 2 glycocalyxes, calyxi, I don't know, that we can have. So remember the characteristics of both, and remember that they are mainly used to protect against phagocytosis. Flagella. So we have several types of flagella, right? Monotrichous, amphitrichous, Dr. B said that she hates this one, but it's supposed to look like if this was the body of my bacteria, that these are the flagella. Like there's going to be a lot on the ends. So that's what Lopotrichus would be. And then Paratrichus, we know that E. coli is Paratrichus, right? It's just going to look like they have long flowing locks. Okay. Amphitrichus is one on each end. Monotrichus is just one on one end. Okay. I don't know to what extent you might need to know this on the exam because I didn't really get that many questions, but she might be changing it up. I don't know. Just be prepared. So understand H proteins as well. I feel like this is an underlooked thing. So H proteins, useful distinguishing serovars. So I'm pretty sure you guys have seen this in probably like a news headline because this is what strain of E. coli can specifically give you a food infection / intoxication. You can get food poisoning from it, and it was recently seen, I think in like 2018, 2017 in lettuce. So that's a fun one, but it's very useful because we can see the variety of species that we have of E. coli specifically. But remember this one, it's kind of just sitting here. I don't know, but I don't know why it's specifically on this slide, but remember motility and our two movements towards favorable conditions. I would also draw this. You'll need to know prokaryotic and probably just review eukaryotic, know the differences mainly. So you see here, flagella is covered in cytoplasmic membrane in eukaryotes, but it isn't in prokaryotes. Know that. The tumble and runs, remember this, that bacteria are not graceful, if you will. And then understand, oh no, I'm gonna edit this on. Understand the mobility of spirochetes. So they have the axial filaments that just they're gonna move like a corkscrew, right? Let's see, flagella, axioclimates. There you go. This is a good little review. So we have prokaryotic versus eukaryotic flagellar movement, ciliary only in eukaryotes, and eukaryotic flagellum up close. And then also know the axial filaments. Know your conjugation pili. That'll be a good one. Pilus, pili. Those are pretty good. We'll go a little bit more in depth with this in the next exam, just so you guys know. Let's see. Remember that the pili is one thing, the conjugation pili is one thing, and then attachment pili or fimbriae is another one. So make sure to do that differentiation. I would relate these back to like a cactus. The whole point of them is that they attach to each other. You know, like how somebody gets stuck with a cactus and it just sticks to them. That's their whole shtick, right? But is attachment. This is a good one. If you would like to copy this down, I would heavily recommend it. Copy it down, add a little bit more depth, maybe. Go back and look into the previous slides and write down anything that doesn't immediately come to mind. But yeah, this is a good review slide. Any of these summary slides, just copy them down and it's a really good little key for you. This is our whole peptidoglycan. I talked about our alternating NAGNAM, a tetrapeptide linked together with a peptide bond. Talked about that. We talked about the difference between gram negative and gram positive. You guys should be pros at the gram stain by now. If not, come in tomorrow to the open lab. I believe there's going to be another one from 4 to 5. I personally won't be there. I was there today, but I have my own class. I'm sorry, guys. But if you guys do have any last minute Monday, Wednesday questions about the exam before Wednesday, let me know. I will answer them as promptly as I can, and I'll try to draw out even things. And then Tuesday, Thursday, you guys are at a little bit of an advantage because you guys have my Wednesday review. So that we can go over it. And then Thursday morning, you guys are even more at an advantage because right before the exam, I have another review session. So there you guys have it. All right. Make sure you know the lipid polysaccharide and the lipid A portion is what makes it toxic. Look, it's nice and highlighted. Make sure you know what the LPS does to people. Oh, and then your atypical cell walls. You have your acid fast bacteria. That's going to be your Mycobacterium and tuberculosis. And then you're going to get your wall deficient ones. What is an exotoxin? Let's go back here. So let me get back to the slide. Okay, so we have our endotoxin and our exotoxins. So I'm just trying to make sure that I tell you guys this. So our LPS is an endotoxin. Okay, an endotoxin is released after the cell dies. So so this endotoxin, yes, right here. So this endotoxin is part of the bacterial cell wall, and it will be released later. So it's inside of the bacteria. Exotoxin, however, is a bacteria that produces a toxin actively. We'll learn about that later. You don't need to know what an exotoxin is quite yet. You'll need to know it for exam 2. So just hold off on it for a little bit. All will come to light, I promise. Just know for this one that LPS is a type of endotoxin. You don't need to know necessarily much more than that. Other than the lipid A portion is what makes it an endotoxin. Okay. So we have our acid fast bacteria. That's going to be our TB, our tuberculosis. And what type of stain do we do for this one? There's a specific name. And I'll go back to that in a second. Okay. I was told that I should actually start calling on people in my I'm sorry. I know people hate it when I call on people, but I was told in my PAL mentoring session that I should start calling on people. So I have a list of everybody who's here, and I'm just going to run my finger up and down them. I don't know what order it's in. I'm just going to pick on one. Daniela, can you tell me what type of stain that we perform on acid fast bacteria? Are you there? You can type it if you want to. Let me make sure that people are here. Oh no, I think she left. Oh, I'm sorry. I just called out the wrong person. All right, let's try this again. Let's go Hannah. You're right underneath on the list. Hannah, do you know what type of stain we do? Okay, so for acid fast bacteria, what type of stain do we perform on acid-fast bacteria? What is the typical stain? If you don't know, it's okay. I mean, it's probably one that you guys haven't looked at in 3 weeks, so it's okay. I can pull it up. Let me go ahead and do that. You guys are seeing the screen. There we go. Let's go down to here. Alrighty, so we have our Zeal-Nelson acid-fast stain. So acid-fast and waxy cell walls should be in the same vein for you guys, okay? Our primary stain is going to be that carbonyl fusion, and our counter stain is methylene blue. You should definitely go over and review what is the name of the stain, what is the primary stain, what is the counter stain, and what exactly are a couple of characteristics. So I really don't like how this is set up, because the primary stain is first, but it's the second on the list. So just make sure you know what the primary and counter stain is for Medieval, for Schaefer-Fulton, and for our Zeal-Nelson. We obviously have practiced gram stain in depth. There is that animation. If you are still having trouble on the gram stain, I would recommend going to watch that and coming to the open lab. Let's see. Let's go to, and we know the difference between basic and acidic and stains, right? So let's see. Our basic stain is going to be able to tell us just plain arrangement, right? We can't tell anything else other than arrangement. It's like it's like fingerprinting, right? We can tell what the fingerprints look like, but we don't know who it is, right? Not until we get some other information. Okay. And then acidic dyes, remember this one. Really, the PowerPoints hold all the information. I know it's kind of hard to figure out what you need to know, what you don't need to know. So if I were to go through here and tell you slides that you do need to know, know this one, this one, this one. Definitely know this one. This is going to be what you need the most on your actual lab part. You'll need this one, not so much, I don't think. I never really answered many questions about this, but it's either spirals or spirochete. Know how to do this in lab. You don't need to know the whole, like you don't need to memorize this, just know how to make one in lab. You need to know what heat fixation does. You can simplify it. You'll need to know this stain, this stain, this stain, and then obviously the gram stain. That's it. Those are going to be your main ones right there. Then let me switch back over to, okay, so that's our acid-fast bacteria. You'll need to know your mycoplasma and your L forms, what makes them special. You'll need to know the antibiotic, it functions with L forms. You'll just need to know these two really. Remember to copy down any summary slide. It really helps. And also just look at this. I mean, you guys have done so much. Also, diffusion seems to be like hard for a few people. And so like, you know, when you're reading something, it's a little bit harder to figure it out. But if you were to be looking at a picture, it's a lot easier. So if we're describing simple diffusion, make sure that you can kind of decipher what it is. Like I know I had a lot of trouble with it, and students in the past have too. Let's see. Just know the different types of diffusion, osmosis, obviously. This is a really, really good review slide. I would recommend drawing this out. I'm sorry if I'm going fast, by the way. I'm just trying to get all that information in. You know what? Let's go ahead. I think this is going to be the end of the, and you guys obviously will probably know what endospores are. And this is what type of stain? Let's just says it right there. Just kidding. Schaefer-Fulton is our spore stain. Copy this down. Differences between archaea and bacteria. So that's kind of like our middle ground. They're not necessarily prokaryote and they're not necessarily eukaryote, but they're more like eukaryote. So how about this, guys? Let's go ahead and take a few minute break. I know it's a little scrambled right now. I'm gonna go back to the review on the different types of media. But let's take, I'm gonna give it till 45. I know it's a little break, but I'm gonna cut you guys early at 5:50. So come back for like the last 5 minutes if you'd like. But I've got you, okay? So I'm gonna end the meeting. I'm gonna kick you guys out, but I'll be right back. You guys come up with the questions you'd like me to answer, okay? I'm sorry, guys.